Hello and welcome everyone. My name is Jonathan Banks and I am a third-year DMD PhD student at the UIC College of Dentistry. Thank you for joining us for this groundbreaking video series, which will showcase some of our exceptional researchers and their pioneering research topics. As a student researcher, I've witnessed firsthand the research excellence of our college, which you will experience today. Now, it is my honor to introduce UIC Distinguished Professor, Associate Dean for Research, and the Department Head of Oral Biology, Dr. Bedran-Russo. Dr. Bedran-Russo will share her expertise in bio-inspired strategies for the development of functional biomaterials. Dr. Bedran-Russo?
Thank you, Jonathan, and hello, everyone. I'm very happy to be here to talk to you today about functional materials that are derived from plants and how we're using them to develop dental adhesives to make the tooth stronger and healthier. So I am going to guess that many of you in this audience had had to go to the dentist to get a dental cavity treated. This is not really a guess. This is statistics. 90% of the world population will experience dental cavities over their lifetime. To treat this disease, dentists remove part of that decayed tissue and use a filling. Now let's take a closer look into the tooth. This is a slice of a tooth that has decayed. As we look closely, we learn that these teeth are assembled in a hierarchical form. This hierarchical form provides the tooth with the strength needed for it to survive over our lifetime. What does that mean? That means that at each different level of scale, the tooth has specific properties that make it very strong. Starting from the left to the right, you will see these different hierarchical levels, starting at the micro scale, going into the micron scale, all the way to the nano length scale. Within those three images, you're seeing collagen. This collagen accounts for about 40% of the composition of the tooth, and it plays key roles in keeping the hierarchical structure. Particularly, the collagen crosslinks, this glue that's part of the collagen, keeps our hierarchical structure of the tooth at check. These collagen crosslinks can be better visualized in this image as these black connectors. Now, when teeth have been affected by decay, these connectors, these collagen crosslinks are diminished, and they have a profound effect not only in weakening the tooth, but on the ability of the fillings that dentists put on the tooth to properly adhere to the surface of teeth. So when I was a postdoctoral student, I became quite interested in collagen chemistry, and I started looking at it into greater depth. And that had allowed me to look differently into the tooth. So I came up with a research hypothesis. Are we able to build more collagen crosslinks in the tooth? And if so, would that make the tooth stronger? In addition to that, if we're able to do those using dental adhesives, are dentists going to be able to use these crosslinkers quick enough as part of their restorative procedure? So I embarked into looking at different chemicals that can be used as collagen crosslinkers, with a particular eye for natural agents. And I ended up stumbling into a class of special plant compounds. These plant compounds, called proanthocyanidins, are part of a large class of polyphenols. They are found in many plants and they can be extracted. In fact, they can be extracted from renewable resources, or we can also find them as byproducts of other industries. Our first proanthocyanidin-rich extract that we worked with were from grapeseed extracts. We utilize these grapeseed extracts to treat the tooth, and to our surprise, we saw a 10-fold increase in the stiffness of the tooth. The strengthening of the tooth not only happened on the decayed tooth, but it also happened on sound portions of the teeth. This was such an aha moment for me. researchers get very excited when their research hypotheses are actually correct. So we knew now that we had a lot of power into these proanthocyanidins. However, we needed to learn more about them and exactly what they did to the teeth. So we partner with experts in plant chemistry to harvest very powerful packs from plants. So currently today, in addition to working with grapeseed extract, we actually obtain these proanthocyanidins from barks of pine tree, from barks of cinnamon tree, and from seeds of cocoa fruit. So we learn many things about these proanthocyanidins, and I would like to share 3 important lessons that we learned from them. First, these proanthocyanidins have 8 monomeric building blocks. Think about these building blocks as Lego blocks. All of them are slightly different. But even at this stage of being a monomeric building unit, it already can coat the surface of collagen and protect the collagen from degradation. The second lesson is that we learned that just like Legos, these separate units of proanthocyanidins can come together to have different shapes and different forms. And the shape and form of these proanthocyanidins matter. Proanthocyanidins with units of 3 or 4 can elicit the most potent effect in interacting with the tooth. The third lesson is that the connections between these units also have an important effect. Basically, these units can come together either through a single bond or a double bond. And we learned that double bonds are more rigid than single bonds. And therefore, these packs that have double bonds have even higher potency. So based on this information, we started formulating adhesive systems that had proanthocyanidins with 3 to 4 units and that were connected by these double bonds. We go back to the lab and we do procedures similar to what dentists would do on your teeth. We first remove the decayed part of the tissue. Then we go ahead, paint the adhesive on the surface of that tooth. And then we go ahead and fill in with a tooth-colored restoration. Now, in the lab, we take a very close look into that interface between the tooth filling and the tooth. And in the middle, as you can see in orange, is our adhesive system that pretty much diffuse very comfortably into the surface of the tooth. And here are some of our findings. On the left, you're seeing an interface done with a standard adhesive. On the right, you see an interface that's treated with a pack adhesive. On the bottom, you see the tooth. On the middle, you see the adhesive, and on the top is the filling. What you're seeing in red fluorescence are actually porous areas within the tooth. And the black areas are non-porous areas. So as you look at this a little closer, you will see that the standard adhesives have far more porosities on that tooth than the interface that has been treated with the packs. What we also learned, looking at that green fluorescence, is that on pack interfaces, there is a lot of new collagen cross-linking being formed. But not only the red fluorescence shows this increase in collagen cross-link, it also shows to us that that interface is stronger and more retentive. The next steps will be to try these in the clinic. For us to be able to try these in patients, we need to further refine their composition to make sure that they have long-lasting effects. Our hope is that these natural dental adhesives will be able to make restorations last longer so our patients don't have to get them replaced as often sometimes happens. Nature is quite powerful. There is so much to learn from it, whether that is from the tooth or the plants. I really hope this story inspires some of you that have innovative ideas to not give up on your ideas. They may actually work, and most importantly, they may have an impact on people's lives. Thank you.